Welcome to the Code of Life. I'm Randine Neal. Back in 2016, a tragedy hit the Vancouver Aquarium that rocked the staff, the city, and the entire province. The death of two of its beloved belugas, Aurora and her daughter Qila, just 9 days apart. As scientists searched for the cause of their deaths, they turned to genomics to help. And that tragedy did spark hope. The answers they found became groundbreaking research that will not only help belugas in the future, but several other animal species as well. And Genomics is leading the way to help scientists at the Vancouver Aquarium and several other organizations as well. Dr. Marty Haulena is the chief vet at the Vancouver Aquarium. He joins us now to talk more about this. Dr. Marty, I hear they call you.
Yeah, or they call me all sorts of things, I guess.
Oh,
Yeah.
do they? Do you prefer Dr. Marty?
Just Marty's.
Okay. I know this is tough for you because I know that you're
Absolutely,
absolutely
yeah.
shattered by this, but can you go back and just tell us the story of Aurora and Qila and how they even came to be at the aquarium in the first place?
Sure. I joined the aquarium in 2006. At that point in time, Aurora had been there for quite some time, and Kela had now been kind of growing up and was, I believe, about 12 years old. And so, again, quickly formed a bond with them. They're, absolutely spectacular animals, as so many animals are, and they're very special in their own way. Kela was the first beluga whale that was ever born at an institution or an aquarium in Canada. So she was quite special. I think she was even the inspiration for Raffi's Baby Beluga song. So, quite well known, both of these animals. Of course, the aquarium is such a centerpiece for the city, and people had grown up with these whales. So in, in 2016, when Qila was reported to be off, I think it was on a Sunday, and then not eating the next day, and then a procedure scheduled for the next day. She died that morning, very, very acutely, very suddenly, and without a whole lot of clinical signs. And then, most scary enough, Aurora started showing the same clinical signs within about a day or two of Qila. So, we sort of rushed in and spent a lot of time trying to diagnose Aurora. Very non-specific signs, sort of gastrointestinal upset. Ran diagnostics like crazy when she was alive. Actually thought we'd turned her around for a little bit, and then she finally passed away of an aortic aneurysm. And so I don't even know whether we could have actually turned her around and then she had an aneurysm, or whether it was all related. But as you say, absolutely devastating. I still think to this day, one of the worst situations I've ever been in. Those 9 days where we were working really hard with Aurora. Obviously, the community knew, tremendous support from the community, lots of people who'd known these animals almost their entire lives, involved in the process, very closely. So when Aurora passed as well, obviously we were quite devastated, and then the mission turned to, why? And we just didn't have a smoking gun. I want to say we spent close to $150,000 plus untold in-kind, volunteer type of diagnostics as well, run by everyone. Samples run in triplicate or quadruplicate, sent to veterinary schools throughout North America, certainly all of our local authorities as well. Human hospitals, Vancouver General Hospital came to us, and their pathologists had a look at everything, and no one could find a smoking gun. And then sort of a collaborative genomic project, UBC, as well as BC Cancer, and then Genome BC, of course, kind of called us and said, have you done some molecular stuff, genomics? And of course, we done, the PCRs and sort of the targeted molecular diagnostics. Also used the University of Florida laboratory, Dr. Jim Wellahan and Tom Walczyk, who are our big genome guys there. So samples have been sent there. But, no, we hadn't run, this sort of mass genomic kind of thing looking for any kind of pathogen. So this group of people, headed by Dr. Stephen Jones at BC Cancer, really kind of took this project in and went gangbusters with it, and sequenced all sorts of tissue, looking for anything and everything. And we had one little green light at one point a virus related to bovine leukosis virus, but unfortunately that was just part of the it was a reagent contaminant, so it didn't lead to anything. But nothing, no significant pathogen at all in a large variety of tissues, which is on its own kind of bizarro. But I guess the silver lining to all that was that the group had processed so much tissue, had done so much sequencing, that they called and said, look, I think we've sequenced the entire beluga genome. Is that worth something? And I went, well, yeah. I mean, that's incredible. First of all, I'm still a kid of the 80s and 90s, so I thought it took 14 years to get a genome, right? I mean, I still remember the cover of Time magazine, human genome sequence, 14 years later. So I was like, what? I had no idea. And the sequencing is so incredible now. And Stephen Jones and his team those guys are incredible. When, later when other universities and institutions, we had University of Illinois, for example, or Northwestern, I can't remember which, call up, and their genomics team went, we'd love to help. Have you looked at that? And he said, yeah, we're working with this guy, Stephen Jones. They went, yeah, you got it covered. You're good. So I was like, yeah, I think we're good. So anyway, the genome came out of it, which was kind of the silver lining to that publication, of course, and kind of the legacy of Qila and Aurora preserved forever, I guess.
And just to finish their story, no cause really was ever found for
No,
their death.
I think, in the end, based on just ruling everything else out, we're left with some kind of toxic insult. So some toxin that either was introduced and cleared by the animals, whether, coming from the water, it was brought into the institution, something that was given to them maliciously, or something that, our own water treatment process inadvertently released. So, and unfortunately, no, we still don't know.
So, as you said, out of that tragedy, for the first time ever, a beluga was completely genomically sequenced.
Like my impression is like really, really well. As Stephen says, this is one of the most complete genomes that's ever been published. I was like, wow, that's pretty cool.
And what type of information will that give scientists in the future?
Well, you can just think of what's happened based on the human genome being published, whether it's looking at genetic diseases and prevention of genetic diseases, whether it's looking at populations of humans from one area of the world that are susceptible to something compared to other populations. That's very important for beluga, where we've got several isolated populations, particularly in the Cook Inlet in Alaska, and then right here at home in Canada, in the St. Lawrence. That's an isolated population. Both those are highly endangered. Why are they endangered compared to healthy populations in Hudson's Bay? Knowing the genome helps a lot with that. For example, the St. Lawrence population is quite prone to cancer. Is that just because they're exposed to something? Is it a completely genetic thing because they've been such a small population for so long? What is it? The genome obviously helps with that. There's a person from the northeastern U.S. Oh my gosh, he's a very famous human dentist. Martin's his first name. I can remember that one. Anyway, he looks a lot at narwhal and looks at narwhal tusk. And the Monodontidae, there's only only two species in that group, and that's narwhal and beluga. So there's as closely related in that group as possible. So he sequenced narwhal, we sequenced beluga, and he can figure out which set of genes makes teeth suddenly go crazy, right? And that's absolutely unbelievable. So all this sort of ecological, health-based, population-based, conservation
even
medicine,
environmental-based, right? Looking at water.
Absolutely,
Yeah.
And that's a whole lot. We can get on to where genomics is going and how sequencing is fast and patient-side sequencing and all that kind of stuff, which is taking this stuff to cloud nine. It's unbelievable.
That collaboration with Dr. Stephen Jones,
right,
from the BC Cancer Agency went even further after he sequenced a beluga. You said that several other species were sequenced after that.
Yeah, this has led now to 4 or 5 year collaboration on any number of different projects. One of his graduate students or fellows, postdocs, Dr. Samantha Jones, not related, I think. Welsh, obviously. But yeah, she helped us out with some cancer in sea otters. So in that process, we published on the sea otter genome, and then looked at lymphoma. And the interesting results there is sea otter lymphoma genetically, as far as the genes that are involved and are triggered and turned on, is much more closely related to human lymphoma than to dog lymphoma, for example. And we were treating this animal like a dog lymphoma because that's our case study, right? And they were like, it's actually a little bit more human related. So that's kind of cool. And yeah, we've sequenced now harbor seal, wolf eel, stellar sea lion, several species, the cancer stuff. In our world, looking at environmental conditions, water quality, has applications in space travel. So folks are really interested in Mars habitats and what does water look like and what's healthy, what's bad water, what's normal microbiome. And then so they look at, aquarium comparisons and long-term recycling of closed aquariums versus our open aquarium, and what's the microbiome of those systems. And so, we can sequence out every organism and look at metagenomics now, right? Looking at, the distribution of organisms across an environment and what organisms rise and fall when environmental factors change, which is awesome. And that went, oh my gosh, don't get me started, but that went down the big sea star mortality event that we were involved with over the last few years.
Did Genomics help solve that?
Yeah. So we worked with Dr. Ian Houston out of the University of Cornell, primarily, who's also a genome guy. And he was looking at the surface environment of sea stars and sea stars that were dying and sea stars that were healthy, and noticed that all of a sudden these anaerobic populations of particularly bacteria just kind of skyrocketed in the animals as they were dying just before they died. So, we think a lot of this has been an environmental change, so ocean hypoxia essentially, so warming water, lower oxygen, and that was a trigger event. There was a virus that was associated, probably not the causative agent, but there's a parvovirus that was associated with the mortality as well. So yeah, as part of our big sea star die-off, genomics came into that as well.
And I know that you didn't sequence the killer whale, but you've used genomics to help solve some issues for the resident killer whales.
Right.
as well.
So now we're getting into that metagenomics kind of stuff, the microbiome stuff, and working with folks that are trying to preserve our local endangered species of killer whale, the southern resident killer whales. JK and L Pod, everyone local knows them. They know them better than I do, that's for sure. So yeah, introduced to that group. Again, I'm more of a clinical veterinarian, a primary clinician, so I get involved on usually an individual animal or disease outbreak investigation kind of thing. But we were called in to help with J50, a juvenile killer whale that was obviously sick, and looking at treatment options and future discussions on how do we monitor a sick killer whale, what sorts of things can we do remotely. And so the genomics comes right back. So we're looking at respiratory tract genomics, what are normal organisms, how did organisms change in a sick whale or a starving whale, and comparing also on the digestive tract, because we can get blowhole samples and we can get fecal samples without really disturbing a whale. So that stuff is, very interesting.
It's fascinating. I know that we're in a time of COVID, Dr. Marty, and the aquarium is closed right now. I hear that it's going to open soon, but how's everyone doing there? How are all the animals doing?
Yeah, I think the animals are doing just fine. As far as they are concerned, it's operations as normal. We're certainly back to doing we kind of postponed routine physicals and vaccinations and stuff for a couple of months just to see where we're going and how long this will last.
Although they did get tested for COVID.
They did a couple, yeah.
Which animals?
So we tested the animals that people work with most closely, the mammals that people work with most closely. So stellar sea lion, fur seal, sea otter, and harbor seal for COVID. Now we haven't gotten results back yet. They had to go to a special sort of veterinary route of things, which takes a little bit longer. So they had to cross the border. And of course, border closures had a little something to do with that. But they're at the University of California, Davis for testing.
But they're all fine so far.
Yeah, no one's shown any clinical signs of COVID yet. No, not as far as the animals are concerned. Yeah.
And I know that you had to put out a bit of a fundraising push, not you personally, but the aquarium in general, because a lot of your funding comes through ticket sales, right? And that has just completely disappeared.
Oh yeah, without a doubt. And just like Science World or CAP Suspension Bridge or whatever, we are visitor driven. I mean, that's our whole. It's not really business as it's a nonprofit but its model is it's funded by ticket sales, people visiting. So yeah, we're very much in trouble, like a lot of zoos and aquariums across North America and well across the world, really. And yeah, quite frankly, in a lot of trouble.
But there is talk of reopening, finding a way, social, physical distancing.
Yeah, a lot of folks involved with that. I've got a little peripheral role, sort of maximizing protection to the animals and also minimizing cross contamination if animals do somehow become a vector for COVID. So yeah, looking at one-way traffic flows and opening up in a limited capacity and hoping for early mid-June for at least a trial opening. So yeah, I think everyone's kind of excited about that. It'll be a new experience, but it might be a kind of a cooler, more intimate experience for visitors as well. So we'll see how it all works out.
And final question I asked you earlier, but not on tape. Have the animals noticed a difference, all 65,000 of them, that there's no more people wandering around?
Yeah, I don't think so. I mean, the folks that work with them primarily are trainers or biologists or husbandry staff, however they want to be referred to. They're so good, and they've taken upon themselves to keep things very interesting and fun. So we definitely have animals wandering around places that we wouldn't normally, So the penguins like to do a daily stroll around the aquarium and stuff. So I think actually for some of the animals, it's just sort of fun and different. I think for a lot of animals, they don't really notice, as far as things that they're really concerned about: food, interaction, being happy, light and cycles, and all that is always all systems go and as normal as possible. So I think most animals aren't noticing. A few are getting a little extra attention, I suppose.
All right. Well, thank you so much for giving us this update
and
turning what was a tragedy into something that was really worthwhile for not only the aquarium, but the entire world.
Oh, well, thank you very much. This has been great.
Thank you.